Welcome back to part 2 of our section on the stomach. We'll continue by looking at gastric secretions. We've looked at hydrochloric acid in the previous section, and now we continue with pepsin. It's a zymogen. A zymogen is a digestive enzyme that's secreted as an inactive protein, and then it's converted to active enzymes by removing some of the amino acid. Here's an example of the gastric gland, where we'll see pepsinogen formed, and how we cleave off a little bit of peptide to reveal pepsin, and then pepsin will go ahead and digest dietary proteins, break them down into their component amino acids. So pepsinogen is secreted by chief cells. Here you can see the hydrochloric acid acts to remove this small yellow piece, leaving the active form of the enzyme, pepsin. Pepsin then will have an autolytic effect. So as some pepsin is formed, it will go back and remove the peptides from more pepsinogen to form more pepsin, and thus digest dietary proteins into shorter peptide chains. The protein digestion is then completed when we enter the small intestine. Gastric lipase is also a secretion. It's secreted by the chief cells also. Gastric lipase, along with lingual lipase, are going to play a little bit of a role in digesting dietary fats. Only about 10 to 15% of fats are digested in the stomach. The rest of our fat will be digested in the small intestine. You can see the small intestine has a big role in absorption. Intrinsic factor is also considered a gastric secretion. It's actually the only secretion that's indispensable and only from the stomach. Intrinsic factor is secreted by the parietal cells, and it's essential to the absorption of vitamin B12 in the small intestine. Without intrinsic factor, it can't be absorbed. The way it works is it binds to vitamin B12, and intestinal cells can absorb the complex of intrinsic factor and B12 by the mechanism of receptor-mediated endocytosis. Digestion can continue if the stomach is removed by gastrectomy, but B12 supplements will be needed by injection, or the B12 would have to be complexed with intrinsic factors so that it can be absorbed. As we age, the production of intrinsic factor goes down. There are also chemical messengers secreted in the stomach. The gastric and pyloric glands have various kinds of enteroendocrine cells, and they produce as many as 20 different chemical messengers combined. Some of those are hormones, so they circulate through the blood and stimulate distant cells. Others are paracrine secretions that will stimulate their neighboring cells. And several of them are peptides that are produced in both the digestive tract and the central nervous system. This class of peptides are called gut-brain peptides. Things like substance P, vasoactive intestinal peptide, secretion, gastric inhibitory peptide, cholecystokinin, and neuropeptide Y. We'll take a look at some of these later. After we swallow a bolus of food, we've activated the swallowing center in the medulla oblongata, which is then going to signal the stomach to relax. As food enters the stomach, it stretches it, which activates a receptive-relaxation response, which is that the bolus of food enters the stomach and the stomach contracts against it, so it resists stretching briefly, but then relaxes to hold more food. Soon, the stomach will show a rhythm of peristaltic contractions, which are controlled by pacemaker cells that are located in the longitudinal layer of the muscularis externa. This will result in a gentle ripple of contraction about every 20 seconds that churns and mixes food with gastric juice and promotes its physical breakup and chemical digestion. These contractions become stronger and stronger so that they reach a maximum in about 30 minutes or so. The antrum of the stomach holds about 30 milliliters of chyme. As the peristaltic wave passes down the antrum, it squirts about 3 milliliters of chyme into the duodenum with each contraction. This allows just a small amount of chyme to enter the duodenum, which allows the duodenum time to neutralize the stomach acid and digest nutrients little by little. If the duodenum is overfilled, it inhibits gastric motility. Typically, a meal is emptied from the stomach in about 4 hours. Less time if it's more of a liquidy meal, and as long as 6 hours if it's a very high-fat meal. Vomiting is forceful ejection of the stomach and intestinal contents, the chyme, from the mouth. There's an emetic center in the medulla oblongata, which integrates multiple muscle actions to reverse the flow of digestive contents. Vomiting can be induced by overstretching of the stomach or duodenum, so by putting too much food in the stomach, forcing too much into the duodenum. It can also be induced by chemical irritants, such as alcohol or bacterial toxins. So when you eat a meal that's contaminated with bacteria, the stomach detects that and will elicit this vomiting response. Visceral trauma can also induce vomiting, intense pain, and psychological and sensory stimuli, which is why we can see something really gross and vomit just from the sight. Vomiting is usually preceded by nausea and retching. So you feel very sick and there's some degree of retching. Retching results from thoracic expansion and abdominal contraction, which creates a big pressure difference that allows the esophagus to become larger. The lower esophageal sphincter relaxes during retching, while the stomach and duodenum contract spasmodically, pushing food up into the esophagus. But in retching, it drops back to the stomach as the stomach relaxes and gets pushed back into the esophagus as it contracts. But it does not get past the upper esophageal sphincter. Usually, retching is accompanied by profuse salivation and sweating, usually accompanied by tachycardia. And even if you don't have physical retching symptoms, you sort of know when you're going to vomit. You get this sweaty salivation, and it's time to run. So when vomiting occurs, the abdominal contractions and rising thoracic pressure force the upper esophageal sphincter open. The esophagus and the body of the stomach relax, and the chyme is driven out of the stomach and mouth by strong abdominal contractions, combined with reverse peristalsis of the gastric antrum and duodenum. Sometimes we vomit with no prior nausea or retching. That's projectile vomiting, and it's very common in babies. Chronic vomiting can cause some serious problems. First of all, very dangerous for our electrolyte and acid-base balance. It can also cause erosion of tooth enamel because of the hydrochloric acid in the chyme. This is one of the signs of an eating disorder called bulimia. Aspiration or inhalation of vomit has some very destructive effects to the respiratory tract. That's why with surgical anesthesia that can induce nausea has to be preceded by fasting so that there are no contents in the stomach and small intestine. Salivary and gastric enzymes partially digest proteins and some starch and some fat in the stomach. But most digestion and nearly all absorption is going to occur after the chyme is passed into the small intestine. The stomach does not absorb any significant amount of nutrients. It will absorb aspirin and some lipid-soluble drugs. But this will explain why drinking alcohol on an empty stomach will give us a buzz much faster, because the fluid moves straight through the stomach and into the intestines. So how is it that we can have such an acidic environment in the stomach and enzymes to break down proteins? How is it that this environment doesn't digest itself? There are three things. First, there's a mucus coat, really thick, very highly alkaline mucus that is going to resist the action of the acid and the enzymes. Second of all, there's tight junctions between each of the epithelial cells, holding the cells so tightly together that it protects them from gastric juice seeping between the cells and digesting the connective tissue in the lamina propria or beyond. Also, there's high rate of epithelial cell replacement. The stomach epithelial cells will only live for about 3 or 6 days, after which they're sloughed off into the chyme and digested with the rest of the food. They're replaced rapidly by cell division in the gastric pits. Breakdown in any of these protective measures can result in inflammation and peptic ulcer. Often stress can result in peptic ulcer. Gastritis is inflammation of the stomach. Peptic ulcers are usually caused by very acid-resistant bacteria called Helicobacter pylori. So most ulcers can be treated with antibiotics and then Pepto-Bismol to relieve the symptoms. So now let's take a look at the regulation of gastric function. The nervous and endocrine systems are going to collaborate to increase gastric secretion and motility when food is eaten, and to suppress them when the stomach empties. Gastric activity is divided into 3 stages called the cephalic, gastric, and intestinal phases. We're going to explore how these phases work by watching the following animation.
Regulation of stomach secretion is divided into 3 phases: cephalic, gastric, and intestinal. In the cephalic phase, the taste or smell of food, tactile sensation of food in the mouth, or thought of food sends nervous impulses to the medulla oblongata. These impulses cause parasympathetic neurons via the vagus nerves to stimulate secretion of hydrochloric acid and pepsinogen in the stomach. The parasympathetic stimulation also results in secretion of gastrin from the lower part of the stomach. This hormone travels through the bloodstream and further stimulates hydrochloric acid and pepsinogen secretion in the upper and middle parts of the stomach. In the gastric phase of gastric secretion, food has entered and distended the stomach. This distension activates a parasympathetic reflex via the medulla oblongata and also has a direct stimulatory effect on the gastric glands. The result is continued secretion of hydrochloric acid and pepsinogen. In the intestinal phase of gastric secretion, chyme has entered the duodenum, so gastric secretion is no longer needed. When the chyme contains lipids from the digestion of fats or contains enough hydrochloric acid to bring its pH below 2, gastric secretion is inhibited. The lipids and hydrogen ions inhibit gastric secretion by 3 simultaneous actions. First, they cause impulses to go to the medulla oblongata to decrease parasympathetic stimulation of the gastric glands. Secondly, they set up local reflexes via neurons in the wall of the gut that decrease gastric secretion. Thirdly, they cause the release of 3 local hormones which travel via the circulation to the gastric glands and inhibit their secretion.
So let's now take a closer look at the positive feedback control of gastric secretion. It begins with ingested food, buffering the stomach acid, that is, making it less acidic. That elevates the pH, and that elevated pH stimulates the G cells. The G cells secrete gastrin, and the gastrin stimulates the chief cells to secrete pepsinogen, and it stimulates the parietal cells to secrete hydrochloric acid. The pepsinogen is converted by the hydrochloric acid into pepsin, and then the pepsin is now an active form of the enzyme, can go and degrade dietary protein. The dietary protein then feeds back and stimulates the G cells directly, and amino acids buffer the stomach acid, making the pH higher again, and they also stimulate the G cell. So now we're in a positive feedback loop, so long as there is ingested food in the stomach. This brings us to the end of our section on the stomach. Next up, we'll be visiting the liver, gallbladder, and pancreas.